Okay, it's 3 o'clock by my clock. Time to get going. Before I introduce Dr. Powell, I have an announcement to make. Remember, next week's colloquium, which is given by Olivier Walther, is going to start at 4 o'clock, not the usual 3 o'clock. I'll say that again at the end today to remind you, and then we'll have it up on the calendar. But today, we bring you Dr. Stephanie Powell from the Department of Geography at the Florida State University. I almost slipped there. There are two purposes of having Dr. Powell on today. One is to introduce people to people from other geography departments in the state of Florida. That's a good, fun thing to do. The other thing is that the sub-theme that I've been kind of pushing this semester, of course, is biologists doing geography and geographers doing biology. There seems to be a convergence of the two fields that are, it's something to do with macrosystems biology, but nonetheless, it's happening. Well, Stephanie is a geographer. She has 2 degrees from UCLA, master's and a PhD in geography, with an undergraduate degree from a really well-known geography program, even though she wasn't, her degrees in environmental sciences from the University of California, Santa Barbara. So Southern California to Florida. She's had a couple, holds a couple of National Science Foundation awards right now. And the good news is she was promoted and tenured last year we're gonna have her around for a long time to come. So without further ado, I'm gonna introduce Stephanie's title and then give the floor to her. This title is Improving Our Understanding of Grassland Ecology and Biogeography Using Neon-Enabled Science. So take it away, Stephanie.
Okay, thanks for that introduction. Let me just share my screen now. All right, and I will minimize that so everybody can see this, I hope. Yeah, so my undergrad degree wasn't in geography at UC Santa Barbara, but I did take all my GIS and remote sensing classes there. So I feel like I was trained in part by their geography department. So today, the title of my talk is Improving Our Understanding of Grassland Ecology and Biogeography Using Neon-Enabled Science. And this is a funded NSF macrosystems biology project that we have. I have all the project collaborators on here. It is the project's really wide in scope, and it's a really collaborative project. So each of us all have our different expertise that we're bringing to it. So I feel like I'm sort of presenting on behalf of the whole group, and then just a special shout out to Dan Griffith, who made, and Bill Riley, who made some of the maps and the slides that I'm using for today's talk. Oh, sorry, just a minute. I can't see my the top of my slides because there's a Zoom menu. How do I move that? Okay, well, I'm just going to
be able to move the Zoom menu over to or away from it.
Yeah, okay, there we go. Sorry about that. I can't minimize it completely though.
You can minimize it completely if you click on the 3 little dots.
Okay, I was just going to do that, but I didn't want to. And then hide video panel.
Controls, yep.
Okay, thanks so much. All right, sorry about that. So I just wanted to start by pointing out that human civilization depends on grasses. So 3 of our most important crops are grasses: wheat, rice, and corn. Grasslands and tropical savannas account for about 40% of the global land surface, about 30% of terrestrial productivity, and are a staple for human sustenance. Grasses are also one of the largest flowering plant families. So Poaceae is a grass family, and it's comprised of about 11,000 different species. But grasses and grassy systems are severely understudied compared to trees and forests, and they're poorly represented in trait databases and in global land models. This has been called the tyranny of the trees. And so here are just 2 recent high-profile papers that sort of illustrate this focus on trees and forests. On top here is a map that's meant to show Earth's tree carrying capacity. So they say that this is mapping tree cover that could potentially exist for climate change mitigation, meaning we should be planting more trees in these areas to sequester more carbon. And the bottom figure is the same idea. They have classified these regions as poorly stocked timberland. And so the problem is that many of these regions are not supposed to be forested. Many of these regions are grassy systems. And since we're in North and Central Florida, we can just think of Florida as an example here. And you can see here much of not just North and Central Florida where we are, but also the coastal plain used to be longleaf pine savanna. And so if we use that as an example, the problem with planting trees in these areas is that comes at a cost to biodiversity and ecosystem function. So in the longleaf, all of the biodiversity is in the understory. So it requires that open canopy environment, high light reaching the understory to maintain that biodiversity. And the other issue is maintaining the ecosystem functions that these grassy systems support. So for example, periodic fires, again in longleaf, periodic fires are required to maintain those ecosystems. So grasses are commonly represented as C3 or C4 plant functional types. Sorry, I'm just wanting to move something. So grasses are commonly represented as C3 or C4 plant functional types. And C3 and C4 are two photosynthetic pathways. 60% of C4 plants are grasses, whereas C3 plants are grasses and mainly woody plants. The C4 photosynthetic pathway is very ecologically successful. So of the 11,000 grass species, about, let's see, I should have this written down, less than 2% of those are C4 species, but the C4 grasses account for about 20 to 25% of terrestrial productivity. So they're hugely successful. So like I said, C3 and C4 are basically two photosynthetic types. Without going too much into their physiology, C4 is basically a warm adapted grasses, and C3 plants tend to occur in cooler environments, and they have an advantage in cooler habitats. And you can see that reflected in their biogeography here. My voice is going because I've been recording so many online lectures for my classes. So you can see that reflected in their biogeography with the C4 plants generally in tropical environments and C3s in higher latitudes here. However, current global land models poorly capture C4 grass distributions inferred from available proxy and plot data. So on the bottom right, what you're seeing is what we know about C4 grass distribution from plot data and from soil and mammal isotope data. And what you can see in these different models is that not only is there poor agreement among models in predicting the distribution of C4, but they also fail to capture what we know about C4 distribution. Much of that gap in our knowledge of C4 distribution reflects a gap in our understanding of C4 traits and how they function. So as an example, the GlopNet plant trait database, there are more than 2,500 plant species in that database. Only 5.3% are grasses or sedges, and of those, only 16 are C4 grasses. So really poorly represented in our trait databases, reflecting our lack of knowledge of their ecosystem function. C4 has evolved independently at least 26 different times in 26 different lineages. And what that means is every time they evolved in a different lineage, they evolved in a different ecological context. So functional trait variation of grasses, and actually of all organisms, results from different evolutionary histories that vary in different ecological and biogeographic contexts. And this results in different ecological behavior and differences in their ecosystem function. So what you're seeing on the left here is an evolutionary tree or a phylogenetic tree. And these are on the bottom here, I think there's 16 primary C4 lineages and then 2 C3 groups here. And you can see that these lineages reflect a very different biogeography. So we can just look at 2 in particular, the Choroidoidae and the Andropogony, which are 2 dominant C4 lineages. And you can see how they're not only in different lineages, but that different biogeographic context is reflected in their distribution here. So on the bottom here, this is a rank abundance curve. And of these 16 C4 lineages and the C3 lineages, 3 of them are globally dominant. And I'm just noticing the gator colors right now. So nice gator colors for this group. So 3 lineages are globally dominant. One of them is a C3 BEP lineage, and BEP is an acronym for actually 3 lineages. Sometimes you'll see me refer to the POEDs, which is the P in that BEP group. And the other 2 dominant lineages based on their global abundance is the Andropogony and the Choroidoidae here. So the Andropogony, this is mapping those 3 lineages in climate space. So mean annual precipitation and temperature here. And you can see that the Andropogony tend to occur in warm and wet environments. And these are grasses that are also adapted to frequent fires. The Choroidoidae tend to occur in warm and drier environments. And then this C3 BEP group, I said that P, the POEDs are the most dominant of that BEP group. So the POEDs tend to occur in cool and dry environments. So our group is proposing that these lineage functional types better capture grass diversity, distribution, and function. Many key aspects of plant function are evolutionarily conserved, or sometimes I'll say phylogenetically conserved, meaning closely related species often have similar traits and niches. Some traits are exceptions, and they'll vary with species, among populations, and over time. And for some of those traits, they might be best modeled through other approaches. So I guess a different way of thinking about what we're trying to do is understand which traits are phylogenetically conserved. So here's a nice side-by-side comparison if we just zoom in on North America, showing how lineage functional types here capture very different biogeography than just a simple C3, C4 plant functional type that's traditionally been used. So our project, again, is really large in scope. There are 3 objectives, and I'm just going to give a quick kind of outline of each of these. And without going into the detail, each one of these objectives sort of has its own set of hypotheses and theory that it's grounded in, but I'll just kind of give a quick overview of each of them. And then for this talk in particular, I'll focus on the second objective most, and I'll talk about what we were able to do this summer because our field season that we had planned was basically canceled. So our first objective is, very simply to, and I'm oversimplifying, very simply to collect an extensive suite of traits for grass species across broad geographic gradients in North America. So physiological, biochemical, life history, structural, hydraulic, and spectral traits. And to understand both how these traits co-vary with each other and whether they sort among lineages, so whether they're phylogenetically conserved. The second objective is to take leaf, so I should say species or functional trait species or functional group spectra, and use those spectra to scale to neon, airborne remote sensing observations, and satellite observations. And this is the part that I'll be spending the most time talking about in today's talk. And the third objective is incorporating lineage functional types into Earth system global land models and comparing those outputs from lineage functional types to traditional C3, C4 functional types, and also using neon products like the eddy covariance productivity estimates as benchmarks for the Earth system model outputs. Sorry, my voice is really going. Okay, so on the bottom is a map of the 7 sites that we propose to collect data at. I haven't talked about NEON yet and just explained what it is for those who don't know, but 1, 2, 3, 4, 5 of our sites are proposed, we've proposed to collect data at NEON sites, and then the others are also LTER sites. So NEON stands for the National Ecological Observatory Network. It's an NSF-funded infrastructure project, and what NEON has done is there are something like 81 sites total, I think. 47 of them are core terrestrial sites. And at every one of these sites, they are systematically collecting a ton of data, everything ranging from, I mentioned the eddy covariance, eddy flux tower data, but also things like plant biomass and abundance, soil moisture, for example, tick and mosquito populations. So a ton of systematically collected data across a very large extent, so across a continental extent. And the LTER sites are also NSF-funded long-term ecological research sites. So our plan was to survey grass species at each one of these 7 sites, and you can see that they captured differences in which dominant grasses are at these sites. We had planned an extensive field season this last summer, but of course that got canceled. So we had planned to go to 3 or 4 of these sites. We'll see about next summer. So what I'll talk about instead is 2 components to my talk today. First, we did collect some preliminary data in 2017. So I'll talk about validating some of the NEON AOP, I'll explain in a bit, but some of the NEON remote sensing data with field-based measurements at Kanza, which is one of our grassland sites. And then we did do some limited field work this summer, again, at Kanza. And I'm happy to talk about all the precautions that we took to do that too. And collected some spectral trait data because the AOP, their airborne remote sensing platform, was flying. So I guess I'll keep you in suspense and tell you if spectral traits sort by lineages. So mainly focusing on this second objective here. So the NEON Airborne Observation Platform, the AOP, which I'll be referring to, has both a hyperspectral sensor and a LiDAR sensor. So this hyperspectral sensor provides reflectance data across 426 bands from 380 to 2500 nanometers. So it's hyperspectral because it's hundreds of bands, very high spectral resolution. If you compare that to something like Landsat, which most people might be familiar with, Landsat has something like 7 bands, 8 bands, 7 of them are at 30-meter resolution, and the NEON AOP is providing data at 1-meter spatial resolution. LiDAR data basically they provide it in both discrete and waveform, but it basically tells you about ecosystem structure, canopy height, for example. And they are flying their sensor over every one of the terrestrial sites, at least on average every 1 to 3 years. So they're providing LiDAR, hyperspectral data, all orthorectified and mosaicked. So there's a lot of promise and excitement in using the AOP for high resolution trait mapping across space and time. And using traits is an approach that should be more generalizable for predicting how species and communities are responding to global change without relying on species identities. So instead focusing on their functional traits, not species identities. The issue is that traits are commonly measured for only a key focal species that represent different ecosystems. And we have limited understanding of how traits vary across space and time. And that's because collecting trait data can be time and labor intensive. So I would argue that remote sensing approaches are actually one of our most promising approaches for understanding how species and communities respond to global change across scales and across environmental gradients, because remote sensing provides continuous observations, complete spatial sampling, what I usually say wall-to-wall sampling, where we don't have to rely on spatial averaging. But for this to be successful, we have to establish rigorous empirical relationships between remote sensing observations and field measured traits. So this first part of my talk, there are two objectives that we had. One was validating AOP-derived data products with corresponding field measurements. And again, this was based on data from 2017, the last time the AOP had flown over Kanza, one of our sites, the last time before this summer. And then the second objective was to examine relationships between the full reflectance spectra as opposed to the products and our field-based measurements. So I'll tell you now that we found poor relationships between the AOP data products and the field measurements. And this work is currently in review at Ecology right now. And our intention is absolutely just to help improve NEON data and to help the research community. The second objective, looking at relationships with the full reflectance spectra, was more promising, but highly variable depending on which trait we were trying to predict. So just to make this point one more time, because sometimes there's confusion about the AOP data. The AOP provides what I'm calling off-the-shelf products. So LiDAR canopy height is one example, or canopy nitrogen. And I just took a screenshot from the NEON website where they offer canopy nitrogen, and you just click download and you get data at a 1 meter resolution for the entire site. Leaf area index is another product that they offer. LAI is basically the area of leaf per unit ground area, and total biomass is another product. So these are derived products from the full reflectance spectra. So we were validating both the derived products, and we also looked at the full reflectance spectra and how well the full spectra could predict different ground-based traits. So and I say we, but actually Jesse Nippert is the PI who led this work with his graduate students. So we as a group, they did really spatially extensive sampling across Kanza Prairie, again, one of our sites, a NEON and an LTER sites, across 200 plots. Kanza is a diverse mesic tallgrass prairie. So it's dominated by C4 perennials, but it's a really diverse mix of C4 grasses, C3 grasses, and also some C3 forbs and some woody plants as well. There are some heavily wooded riparian areas in Kanza, but we avoided those. The other thing that you're seeing on this map, this is a map of Kanza right here, is the different disturbance regimes, so different fire and grazing regimes, showing that it's a really heterogeneous landscape with different resource availability and different disturbance regimes. So we collected all of this, I keep saying they collected all of this trait data in 1 meter square plots. And this is when it was within 2 weeks of the AOP flying over Kanza. And so we took the corresponding AOP data over each one of those 1 meter square plots, but we also took averaged AOP data over a 10 meter circular buffer around each plot, just because the handheld GPS points are usually not accurate to 1 meter accuracy. So these are the traits that we collected. They correspond to the AOP products, leaf area index, canopy height for herbaceous and woody species, total above ground biomass, which was sorted into these different components, but I'll mainly talk about grass and above ground biomass. Those were the largest components. Leaf elemental carbon and nitrogen isotopes. So these are correlations between the derived data products and our field-based measurements. And for canopy height in nitrogen, there was basically no relationship between the AOP product and our ground-based measurements. When you're seeing a lot of R values that are close to zero, so it was like 0.02 correlation coefficients, correlation coefficient of 0.21 and 0.28 starts to look pretty good at that point. So relationships for biomass and LAI were more promising. If we look at biomass first, and this is grass biomass, but it was a really similar R value for total biomass. The first thing you'll notice is that it's off by, the AOP product is off by an order of magnitude. But the second thing is that this is sort of what remote sensors would call a saturating relationship. And that's because their biomass product is based on a spectral index called NDVI. And NDVI is a greenness index, and it's well known among remote sensors to saturate at high biomass values. So for one, the AOP seems to be saturating. It's not able to capture high biomass values. That might in part be because it's based on NDVI. But secondly, the algorithm used to produce these biomass values is based on relationships in forest ecosystems. And so it may be inappropriate to apply that relationship in grass ecosystems or grass habitats. If we look at the relationship with LAI, it's not saturating. There's just a lot of scatter around what's pretty much a linear relationship. So what that means is, on average, if you sample 200 plots across your site, on average, you're going to be pretty accurate 30% of the time. But at any one location, when you're downloading data from the NEON site for each 1 meter pixel, at any one location, it may be completely inaccurate. And a co-author had noticed that we discovered this really interesting relationship. I can't forward my slide right now. The linear lobster and the kicking donkey relationships. And so now that I've seen this, if I go back, I kind of can't unsee it. And all I see is a lobster and a kicking donkey now. All right, so those were the derived data products. We also looked at the full reflectance spectra using partial least squares regression. And these PLSR are common for working with hyperspectral data. So it selects regions of the full spectra. It's a large predictor matrix of 426 bands, and it decomposes them into uncorrelated factors to predict field measured traits. So it's similar to a PCA. That might be the easiest way to explain it. It's not exactly like a PCA though. The number of spectral regions that were ultimately used for the final model, and a comparison of 3 data transformations. These are common data transformations that are used to correct for differences in brightness in the reflectance value, but it also includes just the raw spectra, so doing nothing to it. These were also compared, and the model with the lowest root mean square error of prediction is what I'll report. We split the data into 80% to calibrate a model and then 20% to validate the model. So these were the worst relationships. If we look at, for example, and we're seeing R squares ranging from 0.03 to, what's the highest here, 0.18, right? So these were really poor relationships. If you look at LAI and carbon, the AOP is basically not able to capture any of the variation in field measured LAI and carbon. For some of these, like grass biomass and total biomass, you again see that sort of saturating relationship where the AOP is not able to capture values at the high end of what we measured in the field. More promising were relationships with woody canopy height and nitrogen. So woody canopy height had actually we were pretty successful with an R squared of 0.84. And if I removed this outlier here, it didn't improve the model that much, but this is pretty good already. With nitrogen, it wasn't completely surprising that the models did okay for nitrogen, because nitrogen has been mapped at Kanza before using airborne hyperspectral data, not the AOP sensor, but different sensors. So there's some promise with predicting woody canopy height and nitrogen from the AOP reflectance data. We have been in touch with the NEON remote sensing scientists just to make sure there's something obvious that we're not missing. And the one thing that he did suggest is, because we use that 10-meter circular buffer, he suggested that we use an NDVI filter, because within that wider area, remember our plots were only 1 meter square, but we took the AOP data from a wider buffer. We might be capturing an occasional shadow from a tree or patches of bare ground. So by filtering our data using an NDVI threshold, again, NDVI is this greenness index, then we would be filtering out any pixels that were affected by shadow and bare ground. So we did that, and that did not improve our results. There have been a couple of studies that have successfully used AOP data to map traits. And when you dig into what they did, they're really leveraging much greater variation in traits across sites and biomes. So here, for example, they were able to predict carbon with a much higher R-squared than we were able to. But if you look at their data, they're leveraging much greater variation across sites, whereas we are not looking at just grassy sites, but only one grass site. They had a couple in theirs. With nitrogen, that turnover across these different biomes and sites isn't as obvious, but they still were using 20 times the amount of variation in nitrogen than we were trying to predict at Kanza. So that in part may lead to these better relationships that they're seeing. So just to summarize this first part of my talk, currently available, what I'm calling off-the-shelf AOP products are inaccurate and unreliable, at least at this grassland site. And again, our hope is really just to help improve NEON products. We've been in touch with the NEON remote sensing scientists, but it's also sort of a cautionary tale for those in the research community who might just download data, but aren't remote sensors and may not be aware of the types of pre-processing and data corrections that people do, or be aware of how extensively you do need to validate these data with ground-based measurements. Grasslands may be unexpectedly challenging. So remote sensors and ecologists tend to assume that grasslands are structurally simple, at least compared to forests, but there's actually really high species diversity within a small area that can that can complicate relationships with the airborne spectral data. So within a 1 meter square pixel at Kanza, you might have 8 to 10 different species in just that one small area. Whereas in a forested ecosystem, that would be just one canopy. A few recent studies have had more success using AOP to map traits across biomes, or to map traits, but they're mapping them across biomes, leveraging much greater variability. Or some of these studies have performed extensive data corrections and pre-processing. An example is this paper by Dana Chadwick that just came out. So if you're interested, I encourage you to read that paper. They were in touch with the NEON flight crew, like as they were flying, got preliminary data to choose where they were going to sample. And so what's required is a lot of these pre-processing steps. And the point that we're trying to make is that these aren't, one, readily provided by NEON. Not everybody has access to working with the flight crew that closely. And it also isn't really in the spirit of NEON wanting to serve a diverse research community. NEON provides these data freely and publicly, and there's an assumption that the research community can download and use as is at the 1 meter accuracy. And so again, we're really focused more on the derived products that seem to be inaccurate. And it's in conversation with NEON scientists, it's actually the remote sensing team. It's not a huge surprise to them that some of their products may be problematic. The reflectance spectra, however, was more promising, and developing relationships using field spectra, which we didn't have in 2017, either leaf or plot level field spectra, should improve our ability to map traits. So next I'm going to talk about some limited field work that we were able to do just this past summer. And if anybody wants to talk about the precautions we took before doing field work, I'm happy to talk about that, but lots of testing, quarantining, and that sort of thing. And I have this little note at the bottom because I keep forgetting to say that the reason, why we decided to go was because the AOP last minute decided to fly over Kanza, one of our field sites. And they were not planning to fly for the next few years. So we felt like it was our only opportunity to capture corresponding field data, leaf spectra, for example, with their flight, with their hyperspectral data, with their airborne data. So we went this summer to Kanza with my postdoc Trina Merrick, PhD student Ryan Slapikis, who will be working on this project for the next few years, and Jesse Nippert, collaborator on this project, but also the PI at Kanza, and his undergrad, Ryan Donnelly, who will be joining our team next year as a master's student. we're not sure yet if we'll actually be doing field work this summer, but one way or another, we're all going to be working together at some point in some capacity. So we went to Kanza again this summer, and we were focused on collecting the leaf and plot level spectra. So we built spectral libraries for 32 different grass species, 49 species total, so some of them were shrubs and woody species. And this is a picture of Ryan with our leaf clip and the field spec here. So you kind of put the leaves in a leaf clip and zap them, and you get a reflectance spectra here. And what you're seeing here is that the leaf spectra are sorting by these different lineages. So each of these regions correspond to different traits, different leaf traits. So in the visible region, it's capturing leaf pigments. In the near infrared, you can see differences in the cell structure of these different lineages. The Andropogony really stand out here. And in the shortwave infrared region, it's capturing differences in leaf biochemistry, proteins, lignins, cellulose, water content, for example. So this phylogenetic conservatism, or sorting among lineages in spectral traits, it's been demonstrated before in tropical forests, mainly work by Greg Asner. It's also been demonstrated in the oak family, work by Janine Cavender-Bears, and in broad groupings of seed plants. But we're really excited to see that these grass lineages are also showing sorting in their leaf spectra. So how do spectral traits, one of our questions for this project is, how do spectral traits co-vary with other traits, and do these traits sort among lineages? So this is a table from a recent paper that Dan Griffith led that's out in New Phytologist right now. These are traits that are commonly incorporated into Earth system land models. And I can just give you a few examples. Well, first, we're really excited to fill in this. So we have just preliminary data from one site, but hopefully one day we'll be sampling those other sites and filling this in. But just to give you an example with these two other traits, SLA is specific leaf area, and it basically represents leaf construction costs. So plants might sort along a spectrum of being highly resource acquisitive, and they'd have high SLA values, or highly resource conservative, and they'd have low SLA values. So in general, most models either give C3 plants a higher SLA value, or they assume that grasses are all similar, and they assign one value for grasses. But when we dug into the literature, so these are trait values taken from the literature, what you're seeing is that actually, this is a C3 group here. That was the P and the BEP group that I talked about before. The C3 group actually has lower SLA values than the two C4 lineages. For SLA, there wasn't a significant difference among the two C4 lineages, but if I give you another example of height, here, if you look at height, there's a big difference among these two C4 lineages as well as the C3 lineage. Okay, so for the sake of time, I'm gonna go through a few slides really quickly just to get to some stuff at the end. So really quickly, one of our approaches is to use the leaf spectra to scale to plots based on species abundances, and then to use the AOP to map traits using that plot value. So scaling from leaf spectra to plots, and then using the AOP to map traits. So this is a slightly different approach than just the PLSR with trait values that I showed earlier. This is scaling from the leaf spectra. Secondly, we want to scale to satellite data. So for the sake of time, I'm going to move through these, but what I'll say is the AOP provides really high spatial and spectral resolution data. But if we're able to scale, maybe I'll go back here. If we can scale our data to satellites, that provides not only global coverage or near global coverage with some of these satellites, but it also provides a longer time series. So satellites like Landsat and MODIS, for example, have been providing data for decades at this point. And that can that allows us to look at things like phenology and productivity at a landscape scale. So I'm at 40 minutes. Really quickly, maybe I'll just go through this. So when you look at a Landsat pixel that's 30 meters in resolution, you're getting a mixed signal of different surfaces. And in our case, it would be different lineages. So one approach to unmixing that mixed pixel is called spectral unmixing. And it decomposes reflectance from a mixed pixel into a set of end member spectra. And the unmixing estimates each end member's abundance. So in our case, because we have end members for each of these different lineages, we're hoping to decompose or unmix a Landsat pixel, for example, or a MODIS pixel into abundances of each lineage. Okay, so I'm going to skip forward to here now. Why do we want to scale to satellites? Again, because it can give us a long time series to look at phenology and proxy for productivity over a larger extent and over several decades. So this is a paper from 2014, just to give you an idea of what we can do and plan to do. This is looking at the phenology and a proxy for productivity for C3 and C4 grasses within the same clade, and so controlling for phylogeny. What I did was basically extract an NDVI, I looked at EVI as well, but an NDVI time series based on more than 3,500 digitized herbarium specimens. So 3,500 or more time series that I averaged to estimate an average growing season length for each of these 9 years and an average productivity or a proxy for productivity, which is integrating under the curve basically. What that can tell us is we can look at the relationship between growing season length and productivity. So we looked at these really broad groupings of C3, C4. These are really broad clades. It'll be really exciting to look at this for those lineages that I've been showing. What you're seeing here is that the C3 PACMAD group has the longest growing season length and is the most productive. But the C3 PACMADs are actually globally rare. They tend to occur in warm and wet environments, whereas the most common C3 grasses are actually the poids, and those occur in different environments. So that's one of the things that we hope to look at. But if you just look at this, the biggest difference is between the C3 groups, C3 PACMADs and the C3 BEPs. If you compare the C3 BEP group and this C4 group, what you're seeing is that C3 BEPs had a longer growing season length, but they were less productive than the C4s. So having a longer growing season length does not translate to being more productive. And this is basically suggestive of a slow and steady strategy in resource poor environments. So these BEPs tend to occur in resource poor environments. And it's that resource conservative strategy that I referenced earlier. Okay, so I'll wrap up here. Basic understanding of ecosystem functions in grass dominated habitats have been masked with an emphasis on C3 and C4 plant functional types. But you can see here that lineage functional types capture a very different distribution. Grasses and grassy systems are severely understudied compared to trees and forests, poorly represented in trait databases and in global land models. And that's where we're hoping, that's the gap that we're hoping to fill. And this has consequences for, first, basic ecological understanding of 40% of the land surface. This has consequences for restoration and conservation efforts. So we shouldn't be planting trees everywhere or suppressing fire in many of these regions. And it has consequences for accurate Earth system model predictions that are used in climate-related decision-making and policy. So we're proposing that lineage functional types provide a scalable organizing framework for understanding the diversity, function, and distribution of grasses and grassy systems. And on there, I'm just laughing because my voice is really gone.
Okay, we have plenty of time for some questions. Now, Dr. Powell, sometimes at 3:50 people will bail out because they have a 4 o'clock or 4:05 class. So
yeah, that's why I was rushing, because I think you told me 50 minutes. I can't find the menu to see you guys now, but it's okay.
Yeah, stop sharing. Anyway, so go ahead. If anybody has questions, please jump in. Well, go ahead. Let me ask a question then. You did acknowledge that you had talked to the people at NEON, and a little insider knowledge here is they're planning to suspend the distribution of their derived trait products. That's not widely known at this point, and they've said that at least one investigator has found the poor relationships with things. So I think you just gave me the key for what that's about. But what would happen? You showed that when people do much larger areas and throw in a lot of other vegetation and stuff like that, they start to tease things out. Do you think that will happen with with the other grassland sites too, or will it just still cluster as a kicking donkey?
Right. Yeah, that kicking donkey really sticks with you. So we're hoping that by looking at more variation across sites, we'll be able to capture better relationships. But we're also hoping that starting with the leaf spectra and scaling from there is a better approach to find relationships with the AOP data. So, but just quickly to address, what you were saying about the NEON suspending those products, I had heard that, that they were planning to do that, but there wasn't actually data showing how poor these relationships were as extensively as we had collected, that it was sort of just, I don't know, mutterings and complaints among people.
Well, that's a good question. Have you shared any of your stuff in your paper and review with people like Tristan Golden
Yep, basically Tristan, yeah, because we wanted to make sure what we were doing made sense, and we also, it wasn't meant to be like a hit job on NEON or anything like that, so we were in communication with them, and he's the one that thought it was worth, so it's in review right now, and he thought it was worth publishing because well, I shouldn't say that, but yeah, because there hasn't been an extensive data set showing the poor relationships.
Right. No, I think it's a really good demonstration that the index derived data products really are not very useful.
Right.
That needs to be out there. We don't get enough negative results in the literature that we really need to have. So that's good.
Right.
Yeah. Anybody else have any questions? It's interesting because I just talked about hyperspectral imaging in my remote sensing class yesterday. So several students are taking this class too.
Oh yeah, they're in the audience.
Yeah.
I have a question. This is Sadie.
Hi.
Sadie. So when I've been looking at NEON data for different purposes, I find that the sampling designs are often not how I would necessarily design sampling. And so have you found that the mismatch between different sort of timestamps of sampling across different sites, has that been something you've run into, or is that not featuring in yours?
So do you mean the plot data or the AOP flights?
Basically matching the two is really where it gets funny.
Oh, I see. So that's right. They do their own plot sampling of species abundances, and I don't think they time them to the AOP. They just do those every year, roughly, at the same time. But I think the idea is that at least like plant presence probably shouldn't change too much year to year. But that's why we did our own sampling, and we did it within 2 weeks of the AOP flying. So both the 2017 campaign, but then when we just went in 2020, we were there like the same day they were flying.
Right. So playing off of what Mike Binford was saying, do you think some of that information can get fed back into NEON's design of their own sampling?
I don't think that they will change the AOP flights. So they time those AOP flights based on, a greenness model that they use. And they also have to time them just according to like geography, like at which sites make sense to go next because it's close. I don't know about the plot sampling though. Maybe Mike Binford knows more than us. I have a feeling they try to keep those on a regular yearly schedule, whereas the AOPs are not as regular, and they like to keep it flexible because of weather and other issues.
Well, the AOP is supposed to be flown at a time of peak greenness, whatever that is.
Right.
and that's usually during the time when there are field sampling going on within a few weeks of the AOP flight. But you're right, they don't, that I know of, that they don't actually try to coordinate the two. And you were quite lucky this summer that you were able to get there when the flight was going over.
Well, that was intentional. I got an email from Tristan, and we decided to go. It's like, okay, we can be there in 2 weeks.
Yeah,
2 weeks of quarantine.
but what you said, Stephanie, about the plant presence is not going to change a whole lot from year to year, but the rainfall will, and other kinds of environmental factors will change from year to year, and so there will be some variance that way. But my guess is, now I haven't tried to do the things that you've tried to do, but my guess is that because the AOP flight is done at peak greenness, and that's usually the time when they're doing the field sampling, it will be pretty close in proximity. Not exactly the same day, but pretty close, and which is better than we get in anything else, unless we commission the flights ourselves.
Right. To Sadie's point, though, they also collect a bunch of trait data. I mean, we decided to collect our own, but the traits are probably changing, can change quite a bit within a month or two. So, I think that the NEON data are still pretty new. I mean, I think the flights first started in 2013, but that was just a select number of sites. So it's only been a few years. And I think now you're starting to see people really try to use the data. And that's when these questions start coming up about how useful their design is.
Yeah. Actually, it's 2014.
Yeah, it might be interesting to compare your trait data where it matches the plot trait data if it's offset, and just see sort of how different that becomes.
Yeah, that's true. Yeah.
And they do preserve the samples. They send them out to the biorepository in Tempe.
The physical sample they do. Okay, interesting.
These vouchers.
I had a question, Mike.
Okay.
I enjoyed the talk very much. One of the questions, I guess, that I had in my mind when you're showing the graphs, there was some of the extreme values, some outliers, and I was wondering if there was any retracing the outliers to various samples or locations, and to see if there was any commonalities, or if they're in another context, of something contextual about those outliers that made them outliers. And did you think about at all using other data transformations as opposed to just using Pearson product moment, because that just measures linear associations, but there may be some curvilinear or other types of relationships going on. Did you think about running some of those?
Yeah, so that's exactly right. I'm not sure which traits or graphs you're referring to. One of them, there was a really obvious outlier for canopy height, and that was removed when we did the NDVI filtering, and it did not improve relationships. For some of the others, biomass in particular, it was sort of saturating. And so on the one hand, those few points at high biomass values, you could consider outliers. But that actually speaks to the 2 points you're making. One, maybe those were outliers we could remove. And there was nothing, looking at the data, there was nothing odd. They're not, I'm like hesitant to call them outliers, because they're actually, as far as the biomass values, representative of the site. So there's nothing weird about those high biomass values. But to your point about the correlations being linear, you're right. And so when we plotted it for biomass in particular, maybe a few other traits, it was a nonlinear relationship. I think that the point is, though, again, if you're thinking you could just click download and that you're going to get accurate data at 1 meter resolution, I think our point is still made that it's not that accurate. You can't use
absolutely. That's true. I see that. Yeah. Thank you very much. That was good. Thank you.
Yeah.
Thank you.
Let me interject something here. It's after 3:50 now. The next week's colloquium will be Dr. Olivier Walter, who will be talking about conflict in East and Central Africa. That will be at 4 o'clock in the afternoon, not at 3 o'clock. Olivier has to teach until 4 o'clock. So we'll have him later than that. So it'll be just an hour, it's just an hour later. But we can go back, with that, students turn in your evaluations, and with that, we can go back to questions for Stephanie.
I had a follow-up to Tim's question. I figure you consider this, but Spearman's ranked correlation, like the instead of the Pearson's, in that way to consider those different distributions. I don't know if you did that, but
I didn't run a Spearman rank correlation. It might give us a better R value, but again, I think the point wasn't to get the best correlation we could. It was just to, illustrate that there is a poor relationship between ground-based measures and the AOP. I suppose with the rank, it would tell us if there was at least a relative relationship, but you can see that in the scatter plots that I showed, and there's not really. It's a for some of them at least, there's either a ton of scatter or there is I'm saying a saturating relationship, but another way of thinking about it would be heteroscedasticity, meaning we have much more variation at the low end and then very few points at the high end. And I don't think a Spearman rank would really address that heteroscedasticity.
Probably not. It just, that was a kind of follow up with Tim on that. But thank you.
Sure, thank you.
Cool. Yeah, it's amazing in biology when we see statistically significant relationships with R squareds of 0.2 and 0.3. We're happy with them, but not.
Yeah.
No.
Yeah. Well, if you have a ton of data points, you can get a significance value.
Right.
The poor relationship.
More sensitive. Okay. Well, Stephanie, I think unless somebody else jumps in really quickly, I think we can call it a day. And thank you very, very much. That was wonderful. That gives me a lot of ammunition to take back to NSF, in fact.
Good. Well, again, we're only wanting to be helpful and improve the science. So thank you for inviting me.
One of the things that I guess Sadie's already signed off, but at this point, to change a protocol is a really long, involved set of events have to happen. The technical working group has to chime in on it. The STEAC, the Science, Technology, and Educational Advisory Committee has to weigh in on it. They bring in experts from outside to talk about things. And the field sampling protocols were pretty carefully vetted. There's been a lot of criticism that they're the same everywhere, but that's part of what makes it strong too. And so it's everybody would sample it a little differently, and then nothing would be comparable. So.
That's right. And I think the value of it will be, when we have 30 years of systematically collected data. So, yeah.
But getting these problems ironed out early is important. Actually the observatory has been officially open since May of 2019, just a tiny bit longer than a year. And of course, then COVID hit.
Oh, wow. Okay.
Yeah. And all the data from before that are considered still to be provisional until they come out with their collections. They're going to have an annual collection coming up. So
it's going to be 30 years starting from 2019.
Yeah.
Oh, okay.
So that would be the start date. And the first AOP flights that I know of were 2014. They actually came and did Ordway Swisher was one of the first ones.
Oh, okay.
UF, and then they did, of course, their own site up there near Boulder. So that was, I think, the first one they did. So anyway, cool. So that was excellent. I love that.
Oh, good. Well, thanks so much for inviting me.
Okay, you're welcome. You're very welcome. And back when we're back in